LMU biologists demonstrate how the auxiliary factor CGL160 contributes to the synthesis of crucial parts of the photosynthetic machinery. Photosynthesis is the means by which plants, algae, and cyanobacteria extract their food in the form of energy-rich biomolecules from sunlight, carbon dioxide, and water. It is a complex process, from which researchers are still coaxing many new details. A team led by LMU biologists Thilo Rühle, Bennet Reiter, and Professor Dario Leister has now solved another piece of the puzzle of this essential process and elucidated the role of the auxiliary factor CGL160, as the scientists report in the journal The Plant Cell. Based on the model organism Arabidopsis thaliana, the researchers have now been able to demonstrate that the protein CGL160 plays a key role in the process by recruiting the coupling factor CF1 of the ATP synthase. This portion of the protein binds the CF1 headpiece and facilitates linkage with the portion of the ATP synthase embedded in the thylakoid membrane, making its formation considerably more efficient, explains Thilo Rühle. We observed that the chloroplast structure of corresponding mutants suffers more during short days with just 8 hours of daylight. We advance the hypothesis therefore that the interaction between the N-terminal domain of CGL160 and CF1 is also an evolutionary adaptation that enables plants to cope better in various light conditions, says Rühle. The researchers also found clues as to the evolutionary origin of CGL160. In experiments with Arabidopsis thaliana, they succeeded in functionally replacing the membrane domain of CGL160 with the ATP1 protein from cyanobacteria of the genus Synthesize. Around 80% of the function can be restored in this way. This suggests that CGL160 in land plants may have come from a predecessor protein of cyanobacteria, says Rühle. Indeed, plant cells acquired their chloroplasts through incorporation and functional integration of cyanobacteria. Potential route to regulation of photosynthesis. This set in train the emergence of a complex synthesis plan for photosynthetic machinery. To better understand this plan, the LMU biologists now want to identify all auxiliary factors, in addition to CGL160 and a handful of other currently known factors, that are important for the assembly and function of ATP synthases in plant thylakoid membranes. We assume that there are much fewer factors involved here than in the photosystems themselves. Accordingly, ATP synthase is an easier potential route for the regulation of photosynthesis, says Rühle. In the future, people could use this knowledge for various applications, such as modifying plants so that they can optimally cover their ATP requirements even when exposed to stressors like heat or extreme light conditions. Make a difference. Sponsored opportunity. Story source. Materials provided. Note: content may be edited for style and length. Journal reference: Bennet Reiter, Lee Rosenhammer, Giada Marino, Stefan Geimer, Dario Leister, Thilo Rühle. The Plant Cell, 2022. doi. 10.1093, PL Cell, Coax 306. Cite this page. Ludwig Maximilians Universität München. Photosynthesis. Auxiliary factor ensures efficient energy production. Science Daily. https://ift. tt. w5uxphk. Accessed November 2, 2022. Thanks for watching. Please subscribe my channel for more videos.